Hello friends, how are you today? So here I am again with my one more video on microRNA. MicroRNA or miRNA is the part of RNA interference pathway or gene silencing. Gene silencing is the regulation of gene expression in the cell to prevent the expression of a target gene. And when it is achieved by the RNA molecule, it is known as RNA interference. Many non-coding double-stranded RNAs, namely siRNA, miRNA, shRNA, piRNA, and there are many more, have been found involved in RNA interference. I have discussed the RNA interference and non-coding RNAs in length along with the siRNA in one of the earlier videos in this series. So if you want to know more about the non-coding RNAs and RNA interference, please go through that video so that you have a clear concept of this phenomenon. First miRNA was Lin4 miRNA, which was discovered in 1993 by a group led by Victor Ambrose. miRNAs are endogenous, expressed in almost all the cells of mammals, and these are conserved sequences. So this way they are different from siRNA because siRNA are not present in mammals. Hundreds of miRNA have been identified from plants and animals till date. These are short, around 21 to 25 nucleotide in size, and are partially complementary to target messenger RNA. That means they show imperfect pairing. This feature is also different from siRNA, where the pairing is perfect. Okay, so now we should talk about the biogenesis of microRNA. There are many genes in our genome which encode small RNA molecules, which we call miRNA. They contain the pieces or segments of similar code to other genes. These are double-stranded molecules and they activate RNA interference. The microRNA genes are usually transcribed by RNA polymerase. It binds to the promoter near DNA coding sequence, and after processing in pre-miRNA, it becomes hairpin loop structure. The resulting transcript of miRNA is single-stranded, a large molecule of about 1000 to 2000 nucleotides. Polyadenylation or poly-A tail formation takes place at 3' end, and the capping is done at 5' end. So you can see that in the diagram. This whole process occurs in nucleus. In fact, the initial steps of processing takes place in nucleus, and then the rest part is in cytoplasm. This long-stranded RNA has complementary structure, so it folds back on itself and makes a double-stranded hairpin structure. Now this miRNA precursor is termed as pri-miRNA. Pri is for primary miRNA. Its length is about 100 nucleotides. One pri-miRNA may contain around 1 to 6 miRNA precursors. In next step, this double-stranded hairpin structure is recognized by DROSHA, which is a nuclear protein that cuts RNA. It is RNA's 3 type of enzyme and acts upon double-stranded RNA. There is another protein found in human nucleus, and that is known as DGCR8. Its full name is D-George syndrome critical region 8 because of its association with a deletion disorder, D-George syndrome. In lower animals like Drosophila and Caenorhabditis, instead of DGCR8, another protein is found which is named as PASHA. DROSHA and DGCR8 both are highly conserved in animals, and together they form the microprocessor complex. Within this complex, DGCR8 directs the RNA's 3-domain of Drosha to cut pri-miRNA, and about 11 nucleotides from the base of hairpin are removed, liberating the hairpins. This processing converts pri-miRNA into pre-miRNA. Pre is for precursor. So it is precursor miRNA. It is shorter in length, around 60 to 70 nucleotide long. It has 5' phosphate group and 3' hydroxyl group with 2 nucleotides overhang. It is same as siRNA. siRNA also has 2 nucleotide overhang at 3' and you can check it in the video on siRNA for details. Up to this point, the processing takes place in nucleus and then the pre-miRNAs are exported to cytoplasm for further processing. Another protein named as exportin 5 is involved in the transportation of pre-miRNA. It occurs through nuclear pore complexes and channels in the nuclear membrane. Exportin 5 binds with pre-miRNA by 2 nucleotide overhang and takes it out of the nucleus and then releases it in cytoplasm. It is a process which is energy dependent. From this point, the processing occurs in cytoplasm. Dicer enzyme now gets attached to the pre-miRNA. Dicer, as you all must be knowing, that it attacks on double-stranded RNA as it belongs to RNase 3 family. It cleaves the pre-miRNA, the terminal loop is removed and forms the mature miRNA duplex. But this duplex is imperfect in pairing and is about 22 nucleotide long. At 5' end there is monophosphate and at 3' end 2 nucleotide overhang is there. Both strands are loaded into mi-RISC. RISC is RNA induced silencing complex. Mi-RISC contains dicer, ergonaut and other proteins like TRBP, PECT, helicase, etc. But argonaut is main catalytic subunit of RISC. The proteins may be different in plants and animals. Generally, the strand with lower 5' stability is the guide strand and remains attached to it. The other is called passenger strand. The loose strand will unwind from the guide strand, and this strand means the passenger strand will be cleaved and ultimately degraded. You see, one strand, which is guide strand, is still attached to RISC. This miRNA will act as template to recognize the specific messenger RNA. This miRNA is partially complementary to one messenger RNA or many messenger RNAs. In animals, the complementarity is to a site in 3' UTR, whereas in plants it is for coding region. So wherever there is complementarity, they bind. But remember, this pairing is always imperfect. But in plants, nearly perfect pairing is found. Whatever the case may be, at least 2 to 7 nucleotides should have perfect pairing. Ergonaut cleaves the messenger RNA because it is RNA's H-type enzyme which acts upon single-stranded RNA. In animals, miRNAs mostly inhibit protein translation of target messenger RNA. Sometimes what happens that after the break of messenger RNA, the free guide strand or miRNA again forms the double strand with the messenger RNA, and it will be recognized again because it now becomes double stranded and it will be cleaved. But this happens only sometimes. Mostly the messenger RNA is degraded. Okay, So here it is the overview. You can see the whole diagram in one page. This whole process is divided into two parts. The first part takes place in the nucleus and the second part takes place in the cytoplasm. So let us start from miRNA gene. The RNA polymerase attached to promoter and then it converts the or it transcribes the miRNA, which is single stranded and which is very long. So this folds on itself and this kind of a structure is formed, which is having the hairpin loops, and this is known as miRNA, that is primary miRNA. Then there are two enzymes, DROSHA and DGCR8. These two act upon it and cuts it and converts the pre-miRNA into pre-miRNA. So you can see this is the one pre-miRNA, which is shorter in length. Now this has to be exported to the cytoplasm, and for that you need an enzyme which is known as exportin 5. This is present in nucleus. So now it is exported to the cytoplasm, the same pre-miRNA. So now here you can see this is pre-miRNA which is in the cytoplasm now. Now the dicer is another enzyme which is now present in the cytoplasm. This acts upon this pre-miRNA and the miRNA duplex is formed. It cuts the pre-miRNA, shorter miRNA duplex is formed. Now you need the RISC. This is a complex which have Argonaut, TRBP, dicer, helicase, many proteins are there. Now you see this double-stranded duplex miRNA duplex is attached to it. One strand of it is known as guide strand, the other strand is known as passenger strand. This whole thing is the secondary processing. Unwinding of these two strands will take place and the passenger RNA is degraded. It is not needed. Only the guide strand you can see it is still attached to it. Now this guide strand will now scan the messenger RNA in the cytoplasm, and whichever is having the complementarity that will be attached to it. So here this yellow one you can see this is the messenger RNA, target messenger RNA, and then ultimately this target messenger RNA is either degraded or the translational repression takes place. So that's how the gene silencing or takes place, because the no protein is formed. So that is all about the mechanism or the biogenesis. So this is miRNA pathway of RNA interference. miRNAs have potential to be used as biomarker for the detection of various diseases, and also this miRNA is these days used in therapeutics. miRNAs are generally endogenous, but they can be provided from the outside whenever we are treating the diseases. So that's all about the miRNA pathway. Thank you so much, friends. Keep watching and keep subscribing.